Hi everyone. Great to see so many faces here. We'll be doing a short introduction and click-along tutorial for our tool OpenMMDL, which focuses on protein ligand simulations and also analysis of protein ligand complexes based on OpenMM. So who are we? I'm Niklas, and this is Valerij, and we're from the Freie Universität Berlin, the Molecular Design Lab of Professor Gerhard Wolber. And maybe let's dive directly into what OpenMMDL actually is. OpenMMDL consists of three main components. We have a setup GUI, which just is an interactive interface for setting up your MD simulations. We also have a wrapper for all the OpenMM code, so it's not running, setting up an OpenMM script, but it just is one command you type based on the GUI setup you did beforehand. And after your simulation, we also have an analysis part, which allows you to analyze protein ligand complexes with dynamic pharmacophore analysis. So how do our parts generally work? We start with the setup, which is based around two main pathways. Either if you have a more simple system, you can use our PDB fixer pathway, which is the one we'll be showing today to keep it a bit more simple. Or you can go a bit more into the detailed setup using Amber tools, where we would create an Amber tools script. You could also execute them. After this, you can just call our simulation command, which prepares the system, so solvates it, adds ions, adds a membrane if you want that, and then also conducts the simulation. And afterwards, we do post-processing, which means we center the ligand and protein within the box so that the analysis works cleanly in the end. And finally, we have our analysis portion, where we do pharmacophore-based binding mode analysis, dynamic pharmacophore analysis, which is a concept we developed in our lab, where we work around these point clouds to see distributions of pharmacophore features throughout time. And we also do a stable water analysis to get an idea about how the water molecules move within your simulation. So, how does the setup generally work? As you can see, the Amber preparation part is a way larger portion. We will not be focusing on this today, but if you want to go a bit more into the nitty-gritty portion, you can click through some Amber tools parameters in here, which will allow you to also include more complex things such as cofactors as heme in CYP enzymes, or things like that. So the setup is a bit more in detail. But we also have the PDB fixer preparation path, which just is a simple click along where you just input a PDB and then you can prepare everything in our tool. So what happens after that? The simulation part takes all of your single components, builds them together in one system, execute the MD simulation. You can see during the MD simulation, as was said in the lecture before, the protein often moves throughout the periodic boundary box. So we have to post processes, and you get a simple topology and trajectory file. In our case, most of the time it's a PDB and PCB file. You can then put into an analysis pipeline. And OpenMMDL analysis takes a while until it updates over here. You does stable water clustering, our pharmacophore-based interaction analysis, and some binding mode analysis, also based around pharmacophores. And for those of you who are interested, we can go through a short installation explanation, but it's not that complicated for OpenMMDL. So a prerequisite is that you have a Linux system, as some of the tools we rely on only work on Linux. Therefore, you'd either have to use Linux or Windows subsystem for Linux. If you have a Windows 11 PC, this also works. So you can create a new environment. So then you need a working installation of Conda, because we use Conda to organize our framework. And it works on Python 3.10 to Python 3.12. So 3.13 is not yet tested, and versions before that don't work. So just create a new environment, and then to get the newest version of our tool, you will have to clone our GitHub repository, as we're still trying on implementing it into the Conda Forge thing, but we have some conflicting dependencies with a new interaction and analyzer, which Valerij will be telling you about in a few moments. So just clone the repository, move into your repository, and then just install all requirements through the requirements text and install with pip install dot within the environment. And then everything should work, and you will be able to execute the OpenMMDL framework from your command line, and this is where Valerij will be starting. I'll just show the click along. So if you have your environment installed, you're at OpenMMDL, and then it's just OpenMMDL setup, and this will spawn a locally hosted web frame where you can click through the analysis. And now Valerij will take over for this.
No, it's just need to. Not perfect.
Quick question. So, do we actually need the GitHub repo, or is it okay to just have the CondaForge version?
Basically, right now it would be okay to have the CondaForge. The setup will look a little bit different. The reason is because basically, as Niklas mentioned, we have PLIP that we use for interaction calculations. The second one that I implemented now is ProLib. The reason why we didn't upload the new version to CondaForge is that the creator of ProLib, Cedric, is working in a company, so ProLib is like his part-time work project. So he was able to do the release like 1 week ago. He did the release on pip, so on PyT, but for CondaForge, he didn't have time to basically wrap up everything in the GitHub. So basically, it's like that the new version is in pip, but not on CondaForge, so that in theory, basically, to see ProLib and to see that version of setup, you need to do bit cloning. Also, if you do Conda install OpenMMDL, you would basically have the same functions. The only difference would be that you would have a little bit different interface. Because again, what I added was that I played around a little bit with CSS, so that basically the interface looks now more modernized, more fancy. As you can see, the older version would be a little bit less fancy, but it would basically do the same stuff.
Okay.
So you can still click along.
Yeah. Cool.
But yeah, basically what I'm going to go through right now is let's move that. Okay, yeah.
And I'll take over the clicking for Valerij, and I'll just explain.
Okay, because what we have here is that we have two options. One would be the PDB preparation with PDB fixer. The other would be basically the Ember. The Ember is the standard Ember that you use. So basically, the Ember tools package that would prepare everything for you. PDB is again PDB fixer, and this is the one that we would focus on in this example. And basically, the first option that we would have right here is to select what you want to simulate, which is basically the empty PDB file that you want, and obviously the ligand in that case, that being either SDF or also basically Mo2 that would also work.
So for our tutorial, I uploaded some files in an FU box. The folder within the subfolder files contains the files we want to use for the MD simulation. This is just a version of the new opioid receptor bound to fentanyl directly from the PDB. I prepared it beforehand with a molecular modeling software called MOE, which is not important here, but I want to emphasize that it is important to prepare your protein or PDB complexes beforehand manually. So PDB fixer alleviates a lot of problems with the PDB file. It does some proteination. So the his thing we talked about or was talked about in the lecture previously can be done by PDB fixer. However, it is important that you look at your complex before you feed it into the thing, into PDB fixer, because it does not, for example, model missing loop regions, model unresolved regions you might want to simulate, and it also is not perfect at recognizing proteination states. So if you do this beforehand, you will have a safer input and you will know that everything's correct, or more likely to be correct. So in this case, we prepared the mu opioid receptor. I modeled the loops. I removed some extra structures such as the G protein. And yeah, so we can just click here. I'll add the, maybe Valerij can start talking. I'll add the things.
And basically here then I have the different options for the phosphors. Again, basically most of the phosphors are then available on OpenMM. So that what we do is we check the GitHub repository of OpenMM, what are the XML files that they have there, which I supported, and basically then implement them, because this is what we then run. Basically the backend is then obviously OpenMM. So we check OpenMM and basically OpenFF, and the sense that we use then the CAF versions from there and also the Smirnov. So for this example, we'll take Ember, and basically as you can see, we have CAF and Smirnov. Can you select Smirnov? Yes, for sure. Yeah, and basically for Smirnov, you see that all of the versions, constrained and unconstrained, constrained and non-constrained, depends on basically what items are constrained and not constrained. And here are all of the versions. That one is the newest one, the 3.0. I think there they want to implement also potentially partial charges. At least this was mentioned that they had like partial charges with neural networks. it is quite unstable. So it's like they didn't publish it. So the stable versions would be these ones down below. This is like for experimenting around, testing it, because again, they are still validating it, but it's already accessible for everyone. And basically also have then the minimization, which is the minimization of the ligand per decade. That is an option that you could use. Again, as you know, a decade, if you minimize it, it minimizes the ligand without taking into account the protein. So this is kind of, if you want, you can do it, but again, it can lead to clashes between the ligand and protein, because in that option doesn't take into account the protein. And then the sanitization, this is basically then required for some of the molecules to be then recognized by a decade correctly to be used in the simulation. Because in my cases, I have, for example, QFD that was then required to be sanitized. Some other molecules don't need it. So this is depending on what molecule you simulate.
And the next slide, shortly.
So which would you recommend, like Smirnov or GAF? Like, I mean, general.
I mean, I'm fine. Both are fine, more or less. So basically, I'm kind of using more to GAF, but I tested both, benchmarked both. Both are fine for me. So basically, I didn't see that much difference, to be honest, between those two. So it's kind of, I guess, in that case, more or less a preference thing. Most people would probably use GAF because, again, it's then more used in Ember and so on. So this is the more standard one. Smirnov, they improve it and basically have a lot of papers where they benchmark it, so it's quite good. But it's kind of, in the end, more or less a preference, I would say, which one you would prefer.
It also highly depends on how your ligand is made up. So what atoms are within your ligands, how your ligand looks. So there, as said before, it is best to check the literature, which type of force field works best for your system.
Correct, because same goes also for the water molecules and force that we have here, because as you saw, we had Ember 19, and there were also some papers regarding the new water molecules that were mentioned, like OPC3. Basically, for example, Ember 14 is quite good with T3P, which is the common one, but there were some benchmarking studies regarding Ember 19, and there they mentioned that basically OPC3 is also quite good. So basically, there's also a lot of cases where you, if you're interested, you do literature research to basically see what good combinations there are. I mean, the most common one is probably Ember 14 and T3P, 44P, and the newer one is then Ember 19 with either you still use T3P or you test out the OPC3, which is also a quite more or less new one that is apparently quite good adapted to Ember 19. Correct. So basically here in theory, if you have it on your laptop, you will see here in your view, they have your protein, and here you basically see the protein and the ligands and also all of the other residues, because if you, for example, take it directly from ACSB PDB, then you obviously have different sugars and other additional additives in the PDB, so which you don't want to simulate quite often. You want to simulate the protein and the ligand. So what you can do is basically you can exclude them by clicking on that mark, and then basically their simulation could be developed. It is also quite often the case that, as you saw, what we upload is the PDB and SDF. The reason being that basically we use the SDF, and then the backend use OpenFF to convert it to a specific format, and then basically put it into the topology of the protein, which means that the PDB file that you want to see here should not have the ligand, because else it will be the case you already have a ligand, and then you put a second ligand in the same coordinates, what will lead to clashes, and then the OpenMMDL will complain.
It will crash. It will not only complain, it will crash.
Correct.
Yeah.
So this is why in this case we then exclude the ligand, because again, what will happen is that the ligand from the SDF will be the one that will be basically replacing that one. So what we would usually want here is to only have the protein without anything else.
So in this case, we could keep it because I removed the ligand beforehand and only have the ligand in the SDF file. In this case, we could keep them because it's cholesterols. And cholesterols are, in the case of GPCRs, known as allosteric modulators. So therefore, we could also keep the cholesterols to have a more realistic representation of how the GPCR surrounding in the membrane looks like. Then we could also keep heteroatoms or not. It's a decision you can also make based on what you want to do. The PDB fixer will just handle everything for you in the backend, and you don't need to worry about it.
Correct. And basically here is the next part of us setting up how the environment will look like of the simulation, because right now we were looking at the protein and the ligand, but we will obviously want to simulate it in water and with or without the membrane. so that we can coordinate it, because this is also something that we do in OpenMM to adjust the system for the simulation. And what we can do is add a water box or a membrane.
As we're working with the GPCR, we're going to add the membrane.
Correct. And basically for the water box, if you select just the water box, you can either select the padding, which means basically that you say, okay, 1 Armstrong to the left, to the right, to the up, to the down of the protein. Or you can also do the absolute water box, where you say XYZ should have the amount of waters. For the membrane, it's obviously only the padding. So here, what is happening is you select the membrane type that you want to have, and basically the minimum padding around the protein. And as you can see here, what was also mentioned in the previous talk, is the ionization. So we ionize a system to the default of 0.15 with nothing and chlorate, so sodium and chlorate. Correct. And as soon as we are done preparing that, we see that we have the correct membrane, have the correct settings, we can go to continue.
One small addition. You see a warning up here for the membrane. For placing the membrane, we rely on the protein being centered within the box, and we do this based on OPM coordinates. OPM is a database where you can find PDB structures aligned to a specific space in the Cartesian, to a specific region in the Cartesian space. So the membrane is placed correctly according to how it is within a biological context. So if you are working with membrane-bound receptors or membrane-bound proteins, it is best to retrieve the PDB file you are working with from the OPM database, for example, or again, beforehand, align your structure you have maybe generated with AlphaFold with a similar protein which has been retrieved from the OPM database, to just put it in the right position, because the membrane will not be set in reference to the protein, only the padding will be done. But if the protein, for example, is positioned like this within our box, it will lie within the membrane. And let's think of an ion channel, for example. If the whole of the ion channel is in parallel to the membrane, you will not simulate anything physiological or sensible. So you have to see that it is positioned correctly within the membrane, and this can be done by retrieving everything from the OPM database.
Correct, and basically this is then the last stage of the setup. And as you can see here on the left side, we have the different settings. So since it's done to basically make it a little bit easier for the user, the first one that we will see is the simulation length, where we basically do the simulation in seconds, and obviously in the back we do the amount of steps that we need to simulate. We also show here the calibration length and the platform that you want to use, that being either CPU or you run CPU switch. You will make the simulation probably slow, or you run it with all CPUs. And on the right side, you will also basically see the content file and the script file that is required for the simulation. So as you can see, we have here a lot of different additional setting options. Can you be able to integrate our simulation?
Yeah.
These are then more the detailed ones that were mentioned also in the presentation, that you can have the different pressure, temperature, the step size, the bar step. Here basically we already took some defaults which were in OpenMM setup, which was basically created by the creator of OpenMM, well, the main maintainer currently, Peter Eastman, and basically took them because the idea is that you would already have defaults, and if you don't want to modify them, you would already have a default setting that you could use. If you are interested in modifying them, or basically are more advanced, then you can obviously modify them to adjust them to your needs. So that it already has some pre-defaults, which can be then in the end adjusted. And what you do in the end is basically you do the save all files, which leads to you basically getting the output with, which is a zip, and there you have the script, which is basically that one. We also see again the settings, so if you want to change them afterwards, you don't need to use the GUI again. You can also use the editor and adjust them here if necessary. So you have the script, you have the ligand file that you want to use, and you have the now prepared PDB. So these are then the 3 files that you would have, that you would need for the simulation.
Yeah. So starting the simulation portion is then relatively easy, as you have already prepared everything in your GUI. So let's see where I am here. So you will get these 3 green files in this case, and running the simulation is then just calling OpenMMDL simulation. I'll just show the help. And then you have a few flags you have to give the system to run the simulation. First of all, you need to supply a folder. This is just an output folder where all of the simulation files will be put. So this can be an absolute or a relative path. I just created a folder called 1. So I could set OpenMMDL simulation -f to be ./1. And then we need to specify the location of the script. If we want to start from wherever, the script can be wherever. In our case, it's directly here. So it's OpenMMDL simulation.py. Then we need to specify the topology file, which is just this file. It will end with -processed_OpenMMDL. So you know that is the file which has been prepared through our preparation workflow and is ready to be used. So this will be added to your file. And finally, you add the ligand in form of SDF or Mol2 file. In our case, this was fentanyl. And now I would be able to run it. However, I prepared this on a different system where I had a CUDA GPU, which this laptop does not have. So it would crash anyway. But then you would just execute this command and everything would start running.
Correct. So here a small annotation, because as you can see, this is CLI that was also implemented in the newer version. So if you did only conda install, if you will type that one, you will get an error, which is the reason because basically there we didn't have a whole entry point. For CLI, we had separate entry points in the sense that to run that one, the OpenMMDL simulation, you would need to not have the space, but basically have it like with that underscore. That would be the command that you would use if you only did conda install. Because again, this is now in the newer version, that is not in conda. So basically, that is something that we then also adjusted. Previously, it was 3 different entry points. We kind of had a different command line. Now it's basically one CLI for everything, which is netted and there's a separate command.
Yes, and after your simulation, you will get this prepared few outputs. So we save some checkpoints in case your MD crashes throughout the way, or you hit wall time if you're working on a compute cluster, for example. So you would be able to restart or continue your simulation from the specific checkpoint. You can set up when checkpoints are saved in the preparation step as well. You'll get the input files organized into one folder. You'll get some post-processing intermediate steps saved into a folder. These are not necessary. These can be or become necessary if something went wrong during wrapping. You can go back to these post-processing files and try and start debugging where the error is. The analysis folder I added for the analysis will be looking into in a few seconds. And of course, we will get a final output folder where our wrap trajectories are in. So they're called centered topology.pdb and centered trajectory.dcb. And we have a folder with MD files where just the unprocessed trajectory file that is output during OpenMM is in. So in case everything went well, you can just first look at the output and then think about which files you want to keep or remove. So if everything looks the way you wanted it to in the final outputs folder, you would normally be able to remove the other files. However, in case that doesn't look right, you can always backtrack to previous steps. Yes. So let's go back to the presentation. And now we'll have a look at OpenMMDL analysis, the analysis pipeline we built for looking at your MD simulations afterwards. So this consists of 3 separate parts. As I already said, the stable water interaction, the stable water analysis, the interaction analysis, which is based on pharmacophores, and the binding modes, which is also based on pharmacophores. And as a little deviation from our main OpenMM portion, Valerij will tell you a bit about the packages that are used to set up OpenMMDL. So we use in the backend MD analysis, clip, and prolif, and Valerij will give you a small insights into this.
The clip basically has that on that slide to quickly explain. It's a web server and also a Python package that you could use. And basically what it does is it looks at your PDB files, and then it looks at your ligand, and standardly analyzes the interactions that are present. So as you can see on the website, you would have such an interface with the hydrophobic interactions, different ones, and you will be able to see that on the left side. What it does in the Python package is basically that you get dynamic information, basically these informations in a data frame. So you can basically use a data frame to then start using that to calculate, to create bar plots and other stuff. But basically here, the thing is that it uses TDPs, which was also a little bit tricky for implementing in OpenMMDL, because at least for every frame, you need to write out a PDB, and analyze, because you need to somehow analyze the whole trajectory in the trajectory itself. So you need to convert it also to topology that you then use basically to see the interactions. And this is basically the package that we were using. And we want to, so basically we started now implementing Prolib, because one of the reasons was that the different interaction types, because some interactions and flip were specifically questionable in the sense that the oxygen of a keto group was a hydrogen donor and the appearance of flip, and sometimes the esters were also recognized as having a negative charge. So there were like some specific artifacts which were kind of strange. So I thought, okay, it would be good to have an additional tool to have it, and that would be Coded, which again, as I mentioned, was created by Cedric Doucier, who basically was a PhD student in France and is currently working for Recursion. And that was basically one of the projects that he developed during his PhD. The package is also available on GitHub. Here's the URL for it. Basically, what it does is it uses smart patterns to basically then also analyze the topology and trajectory of your simulation and gives you then the information. Basically, again, for Prolib, it's a bit more complicated because you don't have complete access to the smart patterns. In Prolib, it's a bit easier because it's easier visible in the code what the smart patterns are. So if you want to basically adjust them, you can manually then go in there and say, okay, I don't think that this is a hydrophobic feature. I think that the smart patterns should be like that. And then you modify them and then you have your own version. So it's quite flexible in that sense that you can modify it. And basically, as you can see, it has here the visualization. And the thing that I'm going to focus on in this additional shifter notebook that I made would be the water-bridge interactions, as you can see here. Because to quickly explain why we used Prolib in the beginning and not Prolib, Prolib didn't have water-bridge interactions. So basically, it was having all the interactions except for the water-bridge interactions. And basically, this was a feature that was quite often requested in Prolib, and now in the newest release it was implemented. And as I mentioned, basically the release of the implementation was last week. So it's quite a fresh feature that only is basically now also available in the pip install version. So to install it with Conda, you probably need to wait 1 or 2 weeks until it's then uploaded to CondaForge, and then you can install it and use it. Okay, and basically here's then the Jupyter notebook that I created. We should go explain a little bit about MD analysis and Prolib. So a quick short question. Who is familiar with MD analysis? Okay, so basically to quickly explain, MD analysis is like an overall package for MD analysis, for MD simulations, in the sense that it can do analysis, it can do conversion of topology, so that if you have Amber topology and trajectory, you can load it in and convert it to some other topology. So it's basically quite flexible, and it has a lot of different analysis functions. But this is what I'm going to focus on. And basically, again, so in the Discord, I sent the link to this Jupyter notebook so that you should be able to access it and you should be able to run it. And basically here we only prepared everything, but you can then download and run it on your own PC or basically run it also on Google Colab so that it can be also possible. And basically here, as you can see, start with the installation to basically get the newest version, including the newest version of Prolib. And for the analysis, it was the case that basically it's quite an old package in the sense that it was created maybe in 2010 or 2015. The main maintainer is now Oliver Beckstein, a professor in the US. And basically it was the case that, again, 10, 15 years ago, it was the case that the code was a bit more in the legacy stage. So it was mostly that some parts were added for the analysis and they were included as serial. So it was basically like one after the other. So there was no parallelization, and there were discussions of parallelizing it, but again, legacy code already in there for multiple years. It's tricky to start modifying it. So there was no modification, but in the last couple of years, a couple of people started basically implementing the option to parallelize it with multiprocessing embeds. And basically what parallelization in that case is that you see here, this is then the serial processing. So basically you have a processing that happens one by one. So for example, if you look at the MSD, you have 1000 frames, and you start going that 1, 2, 3, 4, 5. So basically you go to every frame, and for every frame calculate the MSD using only one CPU. But you obviously have on your PC multiple CPUs, so you don't need to only use one. You can basically use multiple ones and say, okay, CPU 1 does frames 1 to 50, CPU 2, 50, 1 to 100, and basically starts picking up to increase the speed. This is what was the idea, and for that one, since it was a legacy code, the base needed to be adjusted a little bit. This was done by Igor Marin, who was basically a PhD student, and in his work in MD analysis, he basically implemented the base, and now it's the case that the base is there, and at some point, the start of the base, the functions were then implemented one by one because it was the case that they were serialized, but they already had the base to be parallelized. So it was the case that basically people started parallelizing them, and since MD analysis is similar to a DKIT open source project, different people could basically then contribute in the sense that, oh, here's a GitHub page, here's the issue, I am going to help now, and basically this is what I did in that case. I saw the issues, I started doing PR requests and implementing the parallelization for different analysis functions. And basically to showcase, here again is a short example from MSD, to showcase that basically this is the MSD, and you can see here the serial, the multiprocessing, and the dust. And as you can see, it's the same result. And as you can see, it also goes up, goes down, goes up, goes down. The reason for that being, I just concatenated the same trajectory. So basically, I used the same trajectory. It's 1, 2, 3, 4, 5 trajectories basically one after the other, copy paste. This is why it looks so strange. The reason being that basically MD analysis has already some time in it that it used for tests, so I just use that, but they are quite short, so I need to make them, to concatenate them to basically have more data to work with. And basically what you would assume if you start using the multiprocessing is that you would have something like a scaling down here. This would be basically something that you would expect, in the sense that if you start now using more CPUs, you would have like a ideal linear increase. That is obviously not always the case, which we will see here, but this is something that you would assume because you think, okay, I have now 50 CPUs, so if I use not one CPU but 50 CPUs, it is 50 times faster, because again, it is then with 50 CPUs. But this is not completely the case, because in some cases, like here, we basically see the performance increase. And as you can see, it's kind of not really going linear. One reason being that it's a Google Colab. So the CPUs here are a little bit questionable. So I'm not sure how well they are. So we see this is more like a showcase. But even if you would run it on your PC, you would see that basically, in that case, the performance would increase not so much. And here it's also the case that here the performance is shown more for how big the simulation is. So basically, if we can go a little bit up again, no, up, no, down.
Here?
Down to, yeah, that's what I need, the MSC speed up.
Oh yeah, so just leave it, yeah?
Yeah. So basically, because as you can see here, down below is the trajectory length, and here's the performance. And the thing is that, again, multiprocessing is quite good with task and multiprocessing, but it takes obviously time to split everything up, put it in the different CPUs and run it, which means that if you have a quite small trajectory, and now you start splitting it up, putting it in different positions, your one CPU would be just faster because it doesn't need to do additional stuff. It basically just directly calculates the MSE and that's it. While here we start splitting up, doing all of the different distributions, communications of the CPUs with each other, so that it reduces the reason to do that. Which means that multiprocessing with task and multiprocessing is good if you have a bigger trajectory. So basically, if you have a quite big trajectory, or if you know that your analysis takes like maybe 20 minutes or 10 minutes, then it is quite reasonable to start parallelizing, or even 5 minutes. But if it only takes like 5 to 10 seconds to parallelize it, to run it with a similar CPU, then it's not really even beneficial or efficient to start parallelizing, because you then just lose in the end a couple of seconds while doing all of this preparation. But as you can see, overall, Again, here the CPUs, I'm not sure how good they are, but even with 2 CPUs, you see that at some point, this is then the zero baseline. At some point, the multiprocessing starts to give you the benefit. And the longer the analysis time will be, and the longer the trajectory, and the more you need to calculate, the better obviously it will be with the parallelization. But for small instances, it's not really beneficial. And if you go down, this is what I mentioned with the CPU and efficiency, because this was also a paper that was done by the lab of Oliver Beckstein, and you basically see the performance of CPUs. Again, this was, as I mentioned, the idea with the linearity, that in theory, this is the performance, and what we should see is that in theory, basically, the more CPUs you do, the faster the speed, that it will basically increase linearly. But it, as you see here, wasn't the case, because they then checked the speed and the amount of CPUs, and they saw that in the beginning the benefit was quite big, but at some point it started getting lower, and the difference between 15 CPUs and 20 CPUs was almost non-existent. The reason being that basically at that step, the problem wasn't the calculation itself, it was the problem, the communication between the CPUs. Because if it's the case that you have one process that needs to wait for the second process to finish, and the first process takes some time, then the second can start, and then the third will realize the second also can start. So that basically in the end, there are so many, they need to wait for each other so long that the benefit disappears. So that you also then, in the end, sometimes don't have the benefit of using too many CPUs, so that you can see quite often for having a good parallelization and a good efficiency is already enough to basically use 8 to 10 CPUs, which would mean that in the end, you basically don't need to occupy your whole PC to run something, so that a couple of them are already enough. And yeah, this is then basically for that one. And as you can see here, it's the case that I've had then a table there with all of the different functionalities that are already implemented or are going to be implemented. So that you can then look at the table and check which ones you can already use and which ones you need to wait a little bit and then until they're implemented. And again, I have the example here with MSD, so you can just adjust it with any other function that you want. And for Prolif, to quickly explain it, basically, as I mentioned, Prolif is a package that is used to calculate protein ligand interactions, and it didn't have the water bridge interactions. So now it was implemented. Basically, the implementation is that you have here then for the fingerprint, which was something that you did before, now the water bridge. Before you didn't have that key, now you have the key water bridge. And what you can also see here is the order. So basically, because in that discussion that me and Cedric had, the idea was that sometimes it's the case that you don't have the interaction of ligand and water. Sometimes it's the case that you have ligand, water, and only then protein. So basically, it's like not directly between only one water standing between the ligand and protein. Sometimes it's multiple waters. And here we thought, okay, it could be reasonable that we add the order. This is something that you can adjust, because depending on your opinion, if it's a reasonable interaction to have multiple orders or not, you can adjust it to your needs. And as you can see here, basically here's how it's calculated. First, we calculate the ligand-water interactions, then the water and protein interactions, and then we connect the water-water interactions. And we go down. And now we basically want to analyze the results. Now go down a little bit. Here's basically how it would look like in a data frame. So as I mentioned, basically it's the case that similar to Blip, we would have a data frame, and here you can see the frame and basically the interaction. If we go a little bit up, we then also see the residues that it's interacting with. So for example, we see here O frame 1, it interacts with threonine and tryptophan. And basically then we can directly use that information in the form of data frame to basically again start working and creating barcodes and other information. Down. And basically here are then the different examples of the visualization, one being the barcodes, where we then here see a little more visible how often and how long they appear, because for example, for that one, for tryptophan, we see that here in a couple of frames it still stays, while for the aspartate it only appears in frame 2 and then completely disappears. And here are the two additional visualization options. They can make it a little bit easier to see that. One is the 2D, where you basically again can see how each is interconnected with each other. So basically that we see, for example, okay, this oxygen is interacting with this water, which is interacting with the second water 370, and that is the one that is interacting with the grouping. And we also quite often then are able to see in that plot the distance. And if you want to see it in 3D, we have also here the 3D visualization, which you should be able to rotate a little bit even.
Rotate? Oh, yeah.
Yeah. And basically here we can then also directly see that how the connection is. That, for example, here we have the water bridge interaction with multiple orders. So that, for example, with that one, the aspartate is only order 1 because it's ligand, that water and the protein. While for that one, it is order 3 because it's the ligand interacts with that water interacts with water 2, the water 2 with water 3, and that interacts with the protein. So that we basically are able to see then water bridge interactions in higher orders, which is also something that Prolif now is able to do, but Prolif was only able to see only in order 1 water bridge interactions. And basically, these are the two main tools and the two additions. Again, the shifter notebook and the Colab notebook that I showed here are available in Discord, so you can download them and use them and basically try it out. Again, both of these tools have quite good documentation and are quite easily available, so you can always click on them and check them out.
Yeah. Now coming back to the main portion of the OpenMMDL analysis, I mentioned 3 parts in the beginning: stable waters, binding mode pharmacophores, and dynamic pharmacophores. Let's start off with the stable water analysis. Why could investigating stable waters be interesting? This slide we already saw in a similar form earlier today. So waters are important for gaining energy. So getting water out of a binding pocket can give us a large energy gain. And how do we find out which waters can be replaced? We can look at the binding pocket and see which waters are trapped within this binding pocket. And this can be done with our stable water analysis, which gives us these water clusters in the end, which show us if a water molecule moves less than a specified amount between specific frames, and then gives us specific clusters of this water molecule. And this can help guide our, for example, pharmacophore design. So we can know, maybe in this region, we can see that we have some polar residues. Maybe we can make some charged interactions. Maybe we can make some hydrogen bonds there to just get this water out of the binding pocket and therefore get an energy gain by freeing the water and therefore gaining not only the energy of the binding through the enthalpy, but we can also get the anthropic gain through removing the water. Then we have this dynamic pharmacophore analysis. What does dynamic pharmacophore in our sense mean? We reduce pharmacophore features down to the point in which the ligand has its interaction feature. And these are reduced down to points, which are then distributed into these pretty point clouds, which show us how the pharmacophore feature moves throughout time. And therefore, we can get an idea of how stable specific features are. But also, for example, if we think about having a cluster here and having a cluster here, and we only see one ligand portion, which maybe sits in the middle between both of those, this can be a hint at that we have multiple binding modes, which we could also adapt to in our design in the end. For example, if we have a long carbon chain that just moves between left and right, we could think about branching this chain and filling both pockets, for example, also to gain a better binding in the end. So then we also have based on our pharmacophores, a binding mode analysis. So you can not only see this in 3D, but we also generate 3D binding modes. And from those, we also generate Markov chains, which means how do specific binding modes change into other binding modes. To get a grasp of how this looks, I have prepared the files, which you also have in the folder. It's in the analysis folder. And we have the binding mode, for example, here. And then we can see the binding mode Markov chains. So in the example I computed, I used fentanyl. This is for the binding modes, maybe not the best example, as fentanyl is a very strong binder to the mu-opioid receptor, and therefore it does not move a lot. However, we can see that there are some minor fluctuations, and therefore we have specific different binding modes. These are generated based on combinations between interactions. So not only is this a hydrophobic feature, but also where and which amino acid is involved in this hydrophobic interaction. And in the end, this allows us to make these Markov chain plots, which let us estimate how binding modes move into each other. So, for example, we plot beginning and end of the MD simulation. So if you did, for example, a docking, and you can see that at the beginning of your MD simulation, you only have a small dot here, and then it goes over to a middle state, which may be a bit larger, and then it goes into another state, which is huge. This might be a hint that your ligand may be a good binder because it stayed within the binding pocket, but your initial binding hypothesis might be wrong. So the ligand moved out, went to somewhere else, and then it maybe flipped and went back into the binding pocket. So this can help us estimate how our ligand moves and how the interactions and how the binding mode changes throughout time. As yes.
Is there associated probabilities with the transitions?
Yes, so the probabilities are there. You can zoom in. This is a very high resolution thing. You can also output it as SVG, but you get the probabilities computed based on frames and based on like probabilities in the end. So you could also do other Markov analysis based on this if you are familiar with Markov chains in general. So back to this. Maybe we'll just go on like this. And in the end, I have a few suggestions how you could use OpenMMDL in a whole drug design workflow. So at the beginning, of course, we have our simulation preparation and our simulation run. However, now you do not need to only feed the output into OpenMMDL analysis. You could also use different tools. I just put 2 examples from our lab. Pyrot, which is also a water-based analysis tool, which gives you apposite pharmacophores based on water interactions. You could also use my new tool, MPath, which does like allosteric communication analysis in your protein. Exactly, so you don't need to only feed it into our output, but also you could use other simulation software. So example, you have GROMEX on your system, but you want to do the dynamic pharmacophore analysis. You could also just use the output of GROMEX as a .grow and .xtc file, input it into our analysis pipeline, and you would get similar results to the ones you would get within our pipeline. So, in the end, of course, every package is not designed by a single person, or 2 people in this case. Valerij and me worked together with our colleague Yu Chen, which focused on the Amber part. So she made the scripts which allow you to prepare your MD simulation through Amber tools. And Leon Ludendorff worked on the water analysis portion. And within our paper, we had a lot of test cases which were supplied by our whole lab. And therefore, we'd also like to thank them. And of course, our Professor Gerhard Feuber. And if you're interested in using any of the tools we develop in our lab, so yeah, allosteric communication analysis, water analysis with Pyrot or OpenMMDL. For your simulations, you can just scan the QR code and you will be led to our GitHub page. And if you have any questions, I'm happy to answer them now. Yeah. Thanks. Any questions, any problems with the setup I can help with maybe? I already got a comment that with the newest GUF thing, seems to not work. You have to change around some numbers. So OpenMMDL is an open source package which we're continuously developing. So I'm also happy if anyone has any bug reports, send them on the GitHub page, send them to me, send them to Valerij. We'll be happy to help you get your system running, and we'll also be happy to fix the change for everyone else. So you can also help make the package better.
So the Markov chain analysis, is that part of the example? In the notebook?
It's also in the example.
The example.
All of the things I show are in the in a form in the example. So the
example. In that Colab notebook.
Oh. Yeah, that's a huge file because there are multiple copies of the MD trajectory because I did not delete the checkpoints. I did not delete the thing, so you can see what the output looks like. That's why it's 10 gigabytes. So you don't have to download everything, but you can. And the computed Markov chain thing is within there. There's also, you would be able to do the 3D visualization of your dynamic pharmacophore, for example, with Ligand Scout. We also implemented a way to output files which are readable by other software. So if you don't like, for example, the way Jupyter notebooks look, like NGL-View looks, you can just open Ligand Scout, for example. And let me see if this works. Yeah, my PC is a bit slow because it's my travel PC. So we can just, no, not this one. Put our complex in here. Oh, this was the big one. Yeah. So my PC is too slow because I forgot to delete the water molecules. So loading protein complexes with water molecules and ligand scout is a bad idea, but just do that. But in general, you would be able to load that in there and then also load this point cloud.pml file. So the extensions aren't shown in Windows 11. I don't know why they do that, but it's a .pml file, which is pharmaceutical markup language. So ligand scout can also read it. You can extract the pharmacophore features, use them for screening, for example. Yeah.
Awesome.
Yes.
So in your simulation of this, you used Monte Carlo barostat and not the typical, I like the Monte Carlo anisotropic barostat. So what's the reason for it?
We just click through the quick preparation.
But usually would you use the
for a membrane bound system, yes. So for a non-membrane bound system, you can use the Monte Carlo. But for a membrane bound system, you're completely right. Yes.
Yeah, great. And I forgot, can you adjust this in the setup?
Yes, there's a drop-down menu and you can choose aerostats or thermostats, everything very easy. Yeah. So something to think about when you prepare a membrane system there. Any other questions? No? Great. Then thank you for listening.
How long is actually the simulation? Because I didn't check.
The simulation is 200 nanoseconds.
200 nanoseconds. Oh, okay.
It's 200 nanoseconds of fentanyl in the more. I removed the protein.
Yeah. Just trying to get an estimate how long it will still run.
Compute.
Yeah, because I'm running it on my desktop at work, and it has a GPU, so maybe it will finish actually.
But it depends what GPU you have. So I computed, I think, 6 hours or something.
Yeah.
I think on my GPU it's quiet, expensive.
Yeah, so OpenMM is also very good at utilizing GPUs. So I don't know, in Gromex, I sometimes have problems getting full GPU utilization. OpenMM, for some reason, it doesn't get faster, but it utilizes all of it.